And our next speaker and the last speaker of the RegSys COSI is Asa Ben-Hur from Colorado State University. And this is another proceedings presentation. He will tell us about comprehensive evaluation of deep learning architectures for prediction of DNA and RNA sequence binding specificities.
Okay. Okay, good evening, everybody. I'm going to tell you a little bit about something that arose out of a class project that I did with some grad students in the fall. So how many of you in the audience have seen or read this deep bind paper? Yeah, I thought so. So a fair number of you, and and a lot of other people did too, and started writing a whole bunch of papers about all sorts of variants of deep convolutional and deep whatever networks. And so I was wondering, well, every one of those papers says that their method works best, of course. Could you really tell which one works the best? Because that's what, our lab is interested in we work in alternative splicing, and of course there's applications of these deep learning technologies there as well, and so we wanted to understand what makes these methods work. And what typically happens in these evaluation studies is that, well, people come up with their own variant of a deep learning architecture. They run their experiments, and then they look at, compare their results to published results. Well, that kind of comparison doesn't really tell me which method works better. It tells me that in our experiments using this model selection strategy, our architecture performed better than this other model. Because, well, we know all these deep learning architectures are pretty finicky, and that how you choose various aspects of the model has a lot to do with the ultimate results. So I had a whole bunch of students in my class, and we decided to run a comparison and try to make sense of what really works. So I'll give a brief overview of the various components. So of course, start with convolution over sequence, and so in convolutional networks, you start with some form of sequence representation. Usually most people use a one-hot encoding. We'll discuss that as well, because that turns out is not always the best thing to do. So what you do in convolutional networks, you have a filter or convolutional filters that you scan your sequence with, much like a PWM, and there's a pooling operation that kind of coarse grains the results until you distill it down to one or several numbers that you then feed to a fully connected layer at the top. So that's just one-dimensional convolution over sequence. Well, a lot of people also have been applying recurrent neural networks, and whereas in convolutional networks everything is treated independently, and only later things are kind of combined together. In recurrent networks, what the network does is it looks at location in the sequence, computes some state, and then it uses that state keeps updating as it scans the sequence until it produces a state at the end that is used for the next layer in the network. And so recurrent networks have been shown to perform really well in natural language processing, and so it's pretty natural to use them also in biological sequences. And, well, two good things. Well, you just put them together and create hybrid architectures that use both convolutional layers and recurrent layers. And in recurrent layers, you can also parse the sequence both ways. You can have unidirectional or bidirectional networks. And the last component that we're going to consider is actually how you represent the sequence. So the standard way is a one-hot encoding, which we all know. But another thing that the bioinformatics community has borrowed from the natural language processing community is word embedding. So word-to-vec, another standard technique that has already shown quite a bit of promise in bioinformatics, is also a good way of representing your sequence. So we're going to, basically going to put all these components together and see what works best. And of course, we used a automatic model selection strategy so that we don't need to bother with setting parameters. So all the architectures use the same model selection strategy, so that the results are the result of changes in the architecture itself. And so some results. So we looked at both ChIP-seq and ClIP-seq compendia of data. So for ChIP-seq data, we used a bunch of ENCODE data sets that were used in the original DeepBind paper. And, well, what we see is that, well, the original DeepBind architecture, yeah, it performs really well, but there are better approaches. So the essence of the results is that typically we see that the more complex architectures work better. So so DeepBind had a single layer convolutional network. Things with a star, that means multiple convolutional layers. So adding more layer gives you a little bit added performance. Now, dilated convolution, that's when your convolutional filters look at larger and larger parts of the sequence. That also helps a little bit. Now, here we start adding, well, in terms of recurrent networks, we looked at both GRUs and LSTMs, more or less same performance. From what I've seen in terms of the differences between LSTMs and GRUs, you need a lot of data for LSTMs to perform better than GRUs. Now, when you add embedding to the mix, it works even better. And if we look at the ClIP-seq data, it's even clearer that the methods that use embedding work the best. So one take-home message is, yeah, if you're going to use deep learning, use some form of embedding, because that seems to, in this case, make even just a regular CNN work almost the same as the more fancier recurrent networks. Now, finally, well, we know that deep learning methods require a lot of data. And so we divided the results by how many training examples we had. And what you see is that if you have more data, then the difference between the more complicated architectures than the simpler architectures becomes more pronounced. Whereas when you have smaller amounts of data, yeah, the more complex methods still perform the best, but it's a very minor difference. Okay, now the reason we apply these methods is because we want to discover, get some biological insight, and not just get good AUCs. And that's where we had a little bit of a surprise that when we went to extract motifs out of the recurrent networks, sometimes things didn't look so good. In fact, yeah, so with just regular convolutional networks, we're all used to seeing nice, pronounced motifs out of the filters that the convolution uses. And it's somehow it just happens that way. We didn't ask specifically, it's not necessarily designed to do that. It's an outcome of what the network has learned. But it seems like the recurrent networks, because they're incrementally scanning the sequence, they learn things quite a different way. They're not necessarily looking for these localized signal as much as the way the convolutional networks do. And so or maybe we need better ways of extracting motifs out of these recurrent networks. We just use the standard technique of looking at what gets strong activations. And also what we've seen is that in the recurrent networks, the activations tend to be more spread throughout the sequence, whereas with the convolutional networks, what we saw is that the activations in the positive examples tend to be at the center of the chip peaks, which is what you would expect. Yeah, this is a part of a little fun exercise I did with one of my former grad students. We wrote a little paper about 10 ways how to fool the masses with machine learning. And deep learning is a great way of doing that. So did we learn something biologically relevant with these sophisticated techniques? So what we did in order to take a look at that is to look at a different assay for the same transcription factors. So for some of the transcription factors, there was both CellX and ChIP-seq data. So in both cases, we tested on the ChIP-seq data, but in one case we trained on CellX, and another we trained on the ChIP-seq data. So in this, these two columns are the original results that we got testing and training on ChIP-seq, and of course results are not as strong with training on CellX and training and testing on chip, but they're still reasonably good. And the more complicated network, the recurrent network that uses a embedding and a convolution and an LSTM, still works better than the simple convolutional network, which to me was even slightly surprising because the length of the sequences was, in one case, it's 100 for the ChIP-seq and 30 for the CellX. So this was definitely good news that these results do generalize quite well. So I hope I've convinced you that it is worthwhile to use these more complex architectures. But if you're looking for motifs, it's not clear that the recurrent networks are the way to go. So with that, I'll take questions.
Hi.
Yes.
Yeah, so you mentioned you used the DeepBind datasets. I was wondering what the negative set was, because in DeepBind they use dinucleotide shuffled data as the negative set.
Yes, that's what we
Okay.
used. Yes.
So just a remark. In Anshul's lab, they found that when you use a dinucleotide shuffled negative set, it actually doesn't work that well on predicting on actual genomic sequences, because the genome is not the negative set. There's not simply a dinuc shuffled version. So I was wondering if you'd considered maybe using the data sets curated in the DREAM challenge, right? So
Yeah, we've not tried
okay,
that.
yeah, and just the other remark is that sometimes we just care about like the high precision region of the predictions because we want really confident predictions. So sometimes AUPRC can give a better sense of like how the model is doing there than just AUROC.
Yeah, these data sets were balanced, so
They were balanced, okay.
Yes, so
All right.
that wasn't a big
okay.
issue there.
All right. Okay.
Yes.
Hi, good evening. I prepared one of the data set that you cited on RNA binding proteins, so sorry for that mess. In all your results, you just show the average across those 31 proteins, while those proteins tend to behave really differently, and some can even be predicted by normal linear regression on k-meres. So did you try to compare the methods by segregating with respect to the proteins?
No, we've not tried that.
Okay. So it would be interesting to see whether any of those methods work on different subgroups in a better way than the others.
Yeah, thank you.
Hi, thank you for the presentation. It's quite impressive that your ECCB SDM model could perform much better than the IDPS in the clip-stack data in RNA protein interaction task. So how did you achieve that? What is the size of the k-mer used for the embedding?
We use a k-mer size of 3, which is actually lower than what we've seen in the literature. And what we've seen, people use more like 4 or 5 or 6, but in some of our preliminary experiments, we saw that actually going all the way down to 3 seemed to perform
is
better.
Pre-trained, or is it trained together with the model?
Sorry?
Word to back?
No, we kept 3 constant throughout the experiment, so we
hard
didn't.
encoding for the 3 more?
Sorry?
Did you use the 3? Did you use one hot encoding for the 3 more?
No, with one hot encoding, it's just that it's at the nucleotide level. The k-mer embedding, you're using k-mers of size 3 to construct the embedding.
That gives you 4 to the power of 3 or possible 3 more?
Well, in that case, it's not a one-hot encoding. The k-mer embedding embeds
you give the
things
count of that mer?
Sorry?
Oh, okay. Also, yeah, get it. So you have the count for that mer?
Yeah.
Okay. So it's not end-to-end learning anymore?
Sorry?
I mean, did you have the order information of the 3 more? I mean, some of the 3 more appears earlier than the other 3 mores.
Well, it's still at each position in the sequence, we have a vector that corresponds to where that k-mer gets embedded. I didn't really explain in detail how the sequence embedding works. Sorry about that.
Okay, sorry, in the interest of time, we have to cut it here for the questions. So before everyone leaves, I would like to thank everyone for attending these great 2 days of the RegSys COSI. I hope you enjoyed it as much as we did. It was really a nice lineup of speakers. And I would like, of course, to thank particularly all the ISMB RegSys track organizers, the organizing committee, So Anaïs Bardet, A. Ferrat, Raluca Gordon, Sean Mahoney, Uwe Oller, Sushmita Roy, and Judith Zog specifically as well. And I hope that we will see you next year for ISMB RegSys in Montreal. Thank you very much.